And we are looking to develop biomarkers and biotypes in drug addiction. In general, you could take any substance use disorder. And
so most of the time in these trials, there is an established disease-related fMRI signature, or there is a classical fMRI sign that is shown on the people that are suffering from whatever the disease is.
But then the question is, okay, when we have a map of brain activations and we know all these networks are activated, how we are going to define the specific outcome measure that is going to be our target as something like a biomarker? How we are going to define which area or which activation plays a better role as something like a biomarker, or in the process of developing biomarkers in the field of fMRI drug reactivity. So there are many questions and challenges.
We wanted to increase the harmonization in the protocols for cognitive and neuroimaging assessment, as well as for cognitive and neurostimulation interventions. And we also wanted to increase the clinical validity of the tools that we generate from neuroscience, and to offer more tools to clinicians that are interested in applying these type of ideas.
So ENIGMA, again, many of you may know this, but it's a shorthand for Enhancing Neuroimaging Genetics through Meta-Analysis. The basic idea is just to give your data a second life, and it speaks to some of the sample size issues that were raised earlier, some of the methodological challenges we have, as raised by Owen. The idea is to combine existing data for secondary meta or mega analysis.
But in reality, at the end of the day, the major challenge in the field is about validity. And when we talk about validity is not going to happen without any sort of collaboration. So we need to have collaboration. And that is the discussion that we started within the ENIGMA addiction group, discussing about, okay, how we might be able to bring fMRI drug reactivity to the ENIGMA addiction working group, and think how we might be able to make a collaborative network of people who are interested to work on a specific area. We put together a checklist of different methodological factors that should be reported if you want to have a transparent reporting. And the checklist is already available as in 2 versions. And as you can see here, 60 colleagues from different parts of the world contributed to this paper. And as you can see here, we still have remain spaces for you guys, that for those who haven't applied for being a member in the consortium. So we are really interested to have you here.
Basically, what you want to report to be totally disclosing, and so people know what you're doing and what the limitations as well as the strength of your data are everything.
That's not surprising that the items regarding the general task information is being classified as important as the items of the participant characteristics and the general fMRI information section.
So what the consensus study, what the experts agreed upon with a very large consensus of above 80%, is that reporting if, when, and how craving is assessed. So the presence, the time point, and the instruments used for craving assessment is essential.
So the first one is abstinence and recent drug use. And this is really important to talk about because several studies, such as the example here by Joe McCrory and colleagues, have shown that brain activation to drug-associated cues is actually higher during abstinence relative to satiety.
Except for an unexpected dip on 2016, we can see that the general trend of publication has been steadily.
But we are interested in extending our database by extracting deeper details, specifically including many of the methodological parameters that were discussed in earlier talks.
So as you can imagine, based on this database that we have, we are going to reach out to those who have published the database and see, as the initial first step, how many people are willing to share their databases and how we can move forward with the next steps and start to plan for the analysis part.